Hi everyone, welcome to our first attempt at a flipped classroom. We're looking at catalytic proteins, the how and why of enzyme action. Everything a cell does depends on the proteins it contains. Proteins are made up of linear polymers called polypeptides. Poly meaning many, peptides is a type of bond. The complete set of proteins produced by a cell is called the proteome. The systematic study of an organism's complete set of proteins is called proteomics. Proteins show great functional diversity. We group them into four major classes based upon their function within the cell. Catalytic proteins, better known as enzymes, control and regulate chemical reactions in cells. Without enzymes, we would not be able to exist as we do, because our reactions would take too long to occur. Structural proteins, they provide support and shape the cell, and they are components of many structures. Mobility proteins involved in the movement of cells, and regulatory proteins, they regulate movement of substances across the plasma membrane. So when you think of protein channels where larger molecules can travel through, they act as signals between cells. They're components of the immune system, which we look at later in the year, and they can be toxins that assist in defense and food capture. So what is cell metabolism? Cell metabolism is basically the sum of all the chemical reactions that take place within an organism in order to maintain growth and normal functioning. So for example, we eat food to sustain ourselves, and then food is broken down in a process of catabolic pathways. If something is catastrophic, we tend to think that it's broken down. So a catabolic reaction is the breaking down of something. So enzymes assist the breaking down of food molecules into useful forms of energy. For us, that's in the form of ATP, lost heat, and many building blocks for biosynthesis. Biological, synthesis, the making of. So the synthesis of biological molecules. So going back to junior science, from what you should have already learnt, all chemical reactions are energy dependent. They'll proceed more slowly at lower temperatures. They'll proceed faster at higher temperatures. So for example, if you conducted a reaction at room temperature, theoretically it would proceed a lot slower than if you increase the temperature to 100 degrees. Cells have evolved complex biochemical pathways to control and regulate cellular processes that sustain life. Enzymes are the key components that assist in this process. Chemical reactions in cells take place in a series of steps called a biochemical pathway. I personally like to think of this in terms of a flow chart. That way I have something that I can visualise when thinking about this process. Each step is controlled and regulated by a specific enzyme and coenzymes. Many enzymes also need cofactors. The difference between coenzymes, cofactors and enzymes is that enzymes are protein-based, whereas coenzymes and cofactors are not protein-based. However, coenzymes and cofactors are required for the reaction to proceed as normal. One of the great things that enzymes can actually do is they lower the activation energy needed for the reaction. So all reactants, so the left-hand side of a chemical reaction, need to have a certain energy before they'll react. So it's like an energy barrier that has to be overcome before a reaction will occur. In biology, we call this the activation energy. All catalysts, enzymes, lower the energy barrier, allowing chemical reactions to occur more quickly. In exams, they love to use graphs. So the graph that is shown below shows the difference in activation energy required with and without an enzyme. What you'll notice is that without an enzyme, you need a much higher level of energy to kickstart the reaction than if you have an enzyme present. One way to think about it, if you had a set of electric beaters and a handheld beater, both of which you were using to beat cream, the electric beater would require a lot less energy from your part to whip the cream than if you're using the handheld beater. In both instances, the cream will be beaten eventually, but the electric beater will be a lot faster. So in terms of biology, the electric beater is like a reaction proceeding with an enzyme. The handheld beater is a reaction proceeding without an enzyme. So an example of an enzyme being used. So for example, digestion. The R groups, so when you think of an amino acid, remember it's the R group that actually determines which amino acid it is. They line the active site and they form molecular interactions with the substrate, so the substance that is entering the enzyme. This weakens the bonds within the substrate and lowers the activation energy required for the reaction, allowing the atoms to be rearranged into products. Enzymes can either build up or break down products. The substrate breaks or is rearranged to form the products of the reaction, and the products are released and the enzyme is able to react again. So the enzyme is not part of the reaction, it just speeds up the reaction. In the picture shown on the left-hand side, this enzyme is using the lock and key mechanism. So what that means is that the substrate is a perfect fit for the enzyme. So for example, your house key to get into your home only fits the lock on your front door. It does not fit into your neighbour's lock. Research into enzymes has shown there may be an alternative mode which is called the induced fit. In this model, the enzyme almost molds itself around the substrate, so it does alter its shape slightly. Either of these models is fine in your exam. They are both accepted at this time. So just following on from the two different types of models, the one shown below, sorry, some of the writings hasn't transferred across into this program well, but the lock and key model, so the shape of the active site is complementary to the shape of the substrate. So it's like a jigsaw piece just snapping in nicely. When the enzyme and the substrate are joined, this is known as the enzyme-substrate complex. One of the things that you'll notice over the course of this year is that complementary shape is a recurring theme in biology. Apologies again, the shape or the picture hasn't transferred across nicely, but so the second model is called the induced fit model. So in this instance, the shape of the active site changes when the substrate binds to it. So over here, we've got our two pieces of substrate that are going to be joining into the enzyme. And what you'll notice is the actual shape of the active site is what changes. Then the enzyme substrate complex is formed and enzyme releases the product. Enzyme can then be reused for future reactions. Okay, so when we talk about the structure of enzymes, so some enzymes act as pure proteins, so they're folded with an active site, so what we call our tertiary structure. One thing that you could do as a form of revision, every time you think about proteins, go through the sequence of what they actually look like in your head. So for example, we've got our primary structure of proteins. So that is just like a chain of beads. The reason I say beads and not something like pearls is that beads can have different colours to represent different amino acids, whereas a chain of pearls, it's more homogeneous, so it's more everything looks the same, so it doesn't actually show you that the amino acids are different. Our secondary structure, we've either got our beta pleated sheets or alpha coils or just your random coils. One thing to remember for the exam, you have to know it's a beta pleated sheet, you have to know it's an alpha helix coil. And finally, thirdly, we've got our tertiary structure where they fold up into a three-dimensional shape. And lastly, when we get our quaternary structure, so we have two proteins that are in tertiary structure joining together. The best example of that is hemoglobin. Some require other factors to become active, so cofactors that are non-protein. So for example, metallic ions. Coenzymes are an organic cofactor. If something's organic, it must contain carbon and hydrogen. For example, vitamins. Okay, so I cannot stress to you how important the next part is. So changing the active site. Changes to the shape of the active site will result in a loss of function of the enzyme. And that makes sense. If you change the actual shape of your lock to your front door at home, your key would no longer fit. So an enzyme is the same. Enzymes are sensitive to various factors such as temperature and pH. Each enzyme has its own optimum temperature and pH. The one thing that it's really important to remember, and this is what kids get caught out on in exams all the time, the optimum temperature for every enzyme is not 37 degrees. In humans, yes, because that is the temperature that our body maintains itself at, but in other organisms, so for example, a carrot would not have an optimum temperature of 37 degrees because carrots don't have a metabolic pathway that will enable their bodies to exist at 37 degrees. Carrots don't have bodies, but you get what I mean. pH is the same. The pH within our bodies varies depending on the location. So the pH of our skin is a lot different to the pH of our stomach that is very acidic. So if an enzyme is exposed to a temperature or a pH above its optimum value, the intermolecular bonds are broken that leads to a loss of function. So the active site doesn't work anymore. If it is below the optimum value, the reaction will either slow or it will not occur. The thing to remember is that below the optimum value, it doesn't denature the enzyme, it just slows down the reaction. An enzyme is denatured when it has lost its characteristic 3D shape, so its active site has been destroyed. So what we've got here is a graph showing 3 different enzymes and their optimum temperatures. Whenever you come across a graph in an exam, it's really important that you read the axes to actually find out what information can you get from this graph. So in this graph, I can actually see that across the bottom, I've got the temperature. Cent, you'd assume would be a prefix for centigrade. And on the y-axis, I've got the rate of reaction, so how fast the reaction is actually occurring at. I've got the 3 different graphs or lines which show me 3 different enzymes and what their optimum temperature is. So, for example, you could assume that the graph in blue, this one, might be something like a carrot because the enzyme operates at an optimum temperature of 4 degrees. The middle one in red, you could probably safely assume that that would be a human or another warm-blooded mammal. So the optimum temperature for that enzyme is around about 37 degrees. And for the last one, we have an optimum temperature of around about 95 degrees. So that might be something like a bacteria that lives in the hot springs. One thing that you may notice with these graphs, so at the peak of the graph, you're looking at the optimum temperature, so where the enzyme performs at its maximum. Just after the peak of a graph is the denaturation point, so when the active site starts to no longer work. You'll notice that the reaction is still proceeding. The reason for that is that when the reaction, say in a human, if we get up to 39 degrees, all of the enzymes won't denature at the same time. So it's a process that some will denature, then a couple of minutes later a few more will denature, and so on. Eventually the reaction will slow, but it will still continue because molecules are always moving. Just without enzymes, they require more activation energy, so the reaction proceeds at a much slower rate. Prior to the optimum temperature, so on the left-hand side of each line, that is when the reactions are occurring at a slower rate, but they are still occurring. So when we look at reaction rates, there's several factors that can actually affect the amount of product made. So first of all, you've got substrate concentration. So how much substrate do we actually have to react? So an increase in the substrate concentrate will increase the amount of product and to an extent the reaction rate. The actual concentration of enzymes, so if I increase the enzyme concentration, I increase the amount of product that I can make per unit time because I've got more active sites that can work at a given time. The other one is inhibition, so things that may slow down the rate of a reaction. One thing to be aware of is limiting factors. So for example, if I've got 100 units of substrate but only 20 enzymes, once all my enzyme active sites are full, that limits the rate of reaction because you have to wait for those enzymes to take in substrate, release product, and then take in the next 20 lots of substrate. Saturation is what that's called, so when all the active sites are taken up, the rest of the substrate cannot be broken down until the sites are free again. So the solution, we need more enzymes to speed up the actual rate of the reaction. There are a couple of different types of enzyme inhibitors. So molecules that are reversible inhibitors, they bind to the enzyme non-covalently. So going back to a bit of the chemistry, a covalent bond, you're sharing electrons. Non-covalent, they're not sharing electrons. And they can dissociate or break from the enzyme. There are 2 forms of reversible inhibitors. Competitive reversible inhibition, so the inhibitor competes directly with the substrate. So in this instance, so we've got our enzyme sitting over here and the active site just here. The substrate and the inhibitor will both perfectly fit into the active site. In this instance, the inhibitor competes directly with the substrate to actually enter the active site. It will eventually break down and the substrate can move in. This is one way that things such as headache tablets work, so they actually compete with the pain molecules to block the signals that tell your body that you are experiencing pain of some sort. The other type of enzyme inhibition is non-competitive reversible inhibition. So reversible, it can be changed back. So in this instance, an inhibitor binds to part of the enzyme but actually away from the active site. Even though it's not actually blocking the active site, it does inhibit the reaction because it won't occur while the inhibitor is still bound to the enzyme in any way. The inhibitor causes a change in the shape of the active site, therefore decreasing the likelihood or the chances of the substrate binding effectively. Our other type of inhibition is irreversible. So once this inhibition occurs, it cannot be undone. So irreversible inhibitors, they bind covalently. So that's a much stronger bond than a non-covalent bond. They bind to the enzyme's active site and they cause a permanent loss of catalytic activity. So basically the enzyme no longer works. Such molecules are toxic to cells. When you think about it, if enzymes can no longer function within a cell, the cell can then not perform its job within the body. Heavy metal cations, nerve gases and natural toxins act as irreversible inhibitors. So once they've taken the place of the active site, substrate can no longer fit in, so their reaction can't proceed at the normal reaction speed that is required by the cell. Okay, just going to give you a quick rundown of protein synthesis. Don't get too hung up on the actual process of this just yet. We do come back to it in quite a lot of detail when we go into genetics, which is part of Unit 3. But just so that you are aware of how it actually works. So within the nucleus, we've got DNA. That's our genetic blueprint or our instruction booklet of how your body needs to operate. The one thing to remember with things like DNA, it's not just the way you look, it's the way that things inside of your body function. So for example, whether or not you can break down things like lactose, fructose, whether your insulin brings down your blood sugar levels, all those type of things are regulated by the DNA that is housed within your nucleus. The reason why we say it's kept in the nucleus, not on the chromosomes, DNA is only visible in chromosomes during a small part of the cell cycle. So you've got your DNA within the nucleus. That gets transcribed into messenger RNA or mRNA. The purpose of the mRNA is that it can actually carry a copy of the blueprint over to the ribosomes. You've been told all through Year 11 how important ribosomes are as part of protein synthesis. So you've also got this thing called ribosomal RNA or rRNA, and its job is to assemble proteins. So it spits out the amino acids in the correct order. The way they get put together is we've got these molecules called transfer RNA or tRNA. They gather the amino acids and then they join them using peptide bonds. When I get a chain of a lot of peptide bonds, I've got a polypeptide, and that makes an actual protein. All of this occurs within the cytoplasm of your cell. The ribosomes, they can be found on your endoplasmic reticulum, so it's the rough endoplasmic reticulum. When you look at a picture of a cell, the one way to differentiate the rough endoplasmic reticulum or normal endoplasmic reticulum from your Golgi, when they both look very similar, is your endoplasmic reticulum is always found close to the nucleus, your Golgi is always found close to the membrane. And that's when you think about what's their actual job, they're going to be located where they need to do their work. Okay, so what we have here is a closer look at the actual process of transcription that occurs in the nucleus. So this basically is where the DNA template strand is used to produce a copy of mRNA. You may have noticed that there's numbers on each side of the DNA strand. So we've got a 3 and a 5. It's called a 3' and a 5' end. Don't worry too much about that. You need to be able to recognise it. The reason, or the way it is named, is based on where the carbon is, but you don't need to understand any of that unless you're doing chemistry. So just be aware that there's a 3' end and a 5' end. When we get into further detail of DNA transcription and translation down the track in Unit 4, then you'll need to know a little bit more about it. But at this point in time, don't stress too much. The one thing to notice though is that the new mRNA molecules or nucleotides that are being added are done at the 3' end. Once the new mRNA strand has been made using the DNA template strand, it leaves the nucleus via the nuclear pores to head over to the ribosomes to be constructed into new protein. So here we're showing the process of translation. What we've already done within the nucleus of our cell is we've transcribed, or we've read the DNA, we've made a template strand, which is mRNA, where I've replaced all of my T's with U's. And one thing that you may notice is that in my mRNA, I've still got my 5' end and my 3' end. So what happens is the mRNA contains a particular sequence of DNA. Every set of 3 is what we call a codon. These big guys that look like giant T's, they're what we call transfer RNA or tRNA. Their job is to collect sets of 3, so codons, and they then put them into a sequence. The actual order of this sequence is determined by your DNA because a given set of amino acids in a certain order codes for a particular protein. So what they do is they translate the message sent by the mRNA into a usable protein. So it's sort of like thinking about we've got our 26 letters of the alphabet that can make billions of different words. With amino acids, even though I've only got 20 different types of amino acid, I can make millions of different types of proteins depending on the number of amino acids, the sequence that they're in, and so on. One thing that you may have noticed about the tRNA molecules is that they are complementary to the mRNA. So again, we use the same base pairing rules. A goes with T, or in this instance, U. C goes with G. So what you can see is that I've got the complementary base sequence being collected. This is just a chart that actually shows you, based on which set of codons that you have, what actual amino acid it is. So the way that you read it, so the letter that is in the first position, you read from here. So for example, I've said, okay, I'm starting with a C. So I need to read, it will be somewhere in this line. Then my second letter is an A, so it's going to be somewhere in this line. My third position is going to be a G, so I've got C, A, and G, so my amino acid is GLN. The one thing to be mindful of in your exam is to actually look at your amino acid chart and see, is it reading DNA or RNA? So depending on if you've got the U's or the T's will depend if you need to transcribe your actual code that you will be given. We will do practice on that in our tute classes. Down the track, when you do actually get to transcribing sequences of mRNA, one way that you can actually see, have I done something wrong? If you get a stop in the middle of your sequence, chances are you've done something wrong. So you'll notice there's a couple of guys that actually code for stop. So if you get one of those in the middle, that is a sign that, oops, I've done something wrong. Go back, check what you've done, and redo it. Okay, well, that's it for enzymes. Hopefully this has helped you. Please feel free to give us feedback. We need your feedback so that we can actually make tools and resources that will help you guys perform to your best. If there's something that you don't understand, please come and see us. Promise we don't bite. And if you need extra explanations or if you want things said slightly differently, come and see us and we'll do our best to help you. See you all in class. Bye.